Hello, everyone. My name is Max Sabelis, and today I'll be discussing this 2014 study that I found entitled Visualizing Cell State Transition Using Raman Spectroscopy. And this paper investigates whether Raman spectroscopy can track the subtle biochemical changes that occur as cells move from undifferentiated to differentiated states. To set the stage, let's talk about why cell state and differentiation are so important. During development and for regenerative medicine, researchers must determine if stem cells remain pluripotent or if they have begun to differentiate. Typically, we rely on genetic markers or staining to check cell state, but these can be destructive or endpoint based. In contrast, Raman spectroscopy is non invasive and is also label free, which provides a vibrational fingerprint of biomolecules such as proteins, nucleic acids, and lipids within living cells. It can potentially give us a single cell resolution readout of cell state without actually killing the cells. And the authors of this paper thus saw an opportunity to examine whether Raman can capture nuanced changes as the cells move from an undifferentiated state towards a specialized cell fate. So, what was the fundamental question Ichimura and his colleagues wanted to answer? Well, they wanted to know if we can detect and visualize at the single cell level the subtle biochemical changes that accompany the cell state transitions as they differentiate, especially in the embryonic stem cells. So, to address this, they chose multiple cell types. They compared well known terminally differentiated lines including fibroblasts, epithelial cells, and hepatocytes, to stem cells, and partially differentiable lines. And by examining the Raman spectra, primarily in the cell nucleus, they hypothesized that they could distinguish varying cell states and illustrate progression from undifferentiated to differentiated phenotypes. And their key premise was that nuclear chemical composition changes, such as protein, chromatin organization, and DNA, are captured by the Raman's characteristic peaks. And therefore, if principal component analysis, or also known as PCA, reveals this consistent pattern, it will indicate that the stem cell differentiation can be monitored label free. To determine whether Raman spectroscopy can capture subtle shifts in cell states, the researchers used a slit-scanning Raman microscope that operated at a wavelength of 532 nanometers. They mainly focused on the fingerprint region of the Raman spectrum, which ranges from about 700 to 1800 inverse centimeters, and it is especially sensitive to signals from proteins, lipids, and nucleic acids. They examined 4 different groups of mouse cells. The first group was embryonic stem cells, or ESCs, which were cultured either with leukemia inhibitory factor, also known as LIF, to maintain their pluripotent state, or without LIF, to allow them to spontaneously differentiate. The second group included terminally differentiated cell lines, such as NIH3T3 fibroblasts, EPH4 epithelial cells, and HEPA16 hepatocytes. The third group was made up of cell lines that can be induced to differentiate. This included Neuro2A cells, which can become neurons, and 3T3L1 cells, which can develop into adipocytes, or fat cells. The fourth and final group consisted of mesenchymal stem cells, or MSCs, which are multipotent and capable of turning into several different cell types. After collecting the Raman spectra from all these cells, they removed the background fluorescence and standardized the data to account for any variation in experimental conditions. They used principal component analysis, also known as PCA, to reduce the complexity of the data by condensing many individual spectral features into a smaller number of principal components. And by plotting the resulting principal component scores, they were able to see whether cells in different states formed distinct clusters, which allowed them to evaluate how well Raman spec could detect the subtle biochemical changes that occur during the differentiation. So let's now discuss Figure 1. The authors show how 3 fully differentiated mouse cell lines, NIH3T3, EPH4, and HEPA-16 produce noticeably different Raman signals in both the nucleus and the cytosol. The top of the figure shows the RGB composite images, where each color represents a specific Raman peak. The blue channel maps the signal around 753 inverse centimeters, which is often linked to cytochrome C. The green channel shows the peak near 1660, which is the amide 1 band, and reflects the protein's secondary structures. The red channel represents the signal at about 2852, which corresponds to CH2 stretching and is a common marker for lipids. One key observation is that HEPA-16 cells show bright red areas and match up with lipid droplets, which are typical of liver cell metabolism. In contrast, NIH3T3 and EPH4 cells have fewer areas rich in lipids. In all three of the cell types, the blue signal highlights where cytochrome C is located, and stronger blue signals may indicate higher mitochondrial density or more mitochondrial activity. Below these RGB images, there are Raman spectra from both the nucleus and the cytosol. They show the important peaks around 1583, which is another marker of cytochrome C, 1003, which reflects the phenylalanine ring breathing, and the 1660 peak, which again marks the amide 1 band. And the nuclear spectra tend to emphasize signals related to proteins and the nucleic acids, while the cytosolic spectra usually show a stronger signal from lipids and cytochrome C. And by showing that each fully differentiated cell type has its own consistent but distinct Raman spectra, the authors show that the Raman mapping can pick up on the unique biochemical fingerprints. And this comparison across the fibroblast, the epithelial, and the hepatocyte lines set an important benchmark, because if these fully mature cells can be clearly distinguished using these spectral patterns, then more subtle changes, like early or partial differentiation, might also be detectable by watching how these same peaks change or shift within individual cells. On this slide, we're going to talk about different cell lines, but cell lines that haven't fully committed to a mature state yet. These include Neuro2A, which is a neuroblastoma line, and 3T3L1, which is a preadipocyte line. And before they are induced to differentiate, Neuro2A cells typically have a rounded, undifferentiated cell appearance, while 3T3L1 cells are small and resemble fibroblasts. Once differentiation is triggered, Neuro2A cells begin extending neurites, which are the branching structures typical of neurons. And this gives them clear neuronal characteristics. At the same time, 3T3L1 cells start accumulating large lipid droplets as they begin shifting into adipocyte or fat cell identity. And after collecting the Raman maps of these cells, specifically focusing on their nuclei, the authors observed that certain peaks in the Raman spectra changed in intensity as the cells differentiated. These included peaks around 1583 inverse centimeters, which is associated with cytochrome C, and 1660, which corresponds to the amide 1 band and reflects protein secondary structure. The authors note that changes in the cytochrome C signals can reflect shifts in mitochondria, which often occur when a cell's energy needs to change as it specializes. Next, they used principal component analysis to analyze the nuclear spectra. And while the spectra from undifferentiated and differentiated cells did show some overlap, because not all cells go through differentiation at the same pace, there was still a clear shift in how the populations clustered in the PCA space. And this shift suggests that a consistent change is taking place in nuclear biochemistry. That might include things like changes in chromatin organization, differences in protein expression, or redistribution of mitochondria, all of which Raman spectra is able to detect. Moving on to figure 4, we move on, or we move on from induced differentiation to a focus on embryonic stem cells, or ESCs, which are fully pluripotent. And in this part of the study, uh, ESCs were cultured with leukemia inhibitory factor, also known as LIF, to keep them in a naive state where they form tight, round colonies. Then LIF was removed so the cells could spontaneously begin differentiating over the course of about a week. And Raman maps of the undifferentiated ESCs show here that relatively uniform distributions in the nucleus and low levels of lipid association signals. But after the 7 days without LIF, the cells appear flatter and showed stronger peaks related to the lipids. And this suggests that some of the cells were beginning to take on identities from lineages when lipid metabolism plays a more active role. Importantly, they also compared the ESC spectra to those of mesenchymal stem cells, or MSCs. The Raman fingerprint of MSCs particularly overlapped with the cluster of differentiated ESCs, and this indicates that once the ESCs begin losing their pluripotent state, their nuclear composition gradually becomes more similar to that of other stem-like cells. And taken together, these findings show that Raman spectroscopy can detect a full range of differentiation states, from early and partial transitions to nearly mature ones. And it can do this without using any labels or markers, just simply by detecting small but meaningful changes in the spectral features of the nucleus. So on this slide, we're going to be looking at figure 6, which I think is the most revealing data set of the entire study, which is a detailed time-based look at how embryonic stem cells go through differentiation over the course of 2 full weeks. The authors cultured embryonic stem cells without LIF and took Raman measurements of each cell's nucleus on days 0, 3, 7, 10, and 14. And they used these measurements to generate principal component, or PC, score plots that map how the cells progress over time. First of all, at day 0, the cell population is highly diverse. In the PCA score plot, the data points are widely scattered, which shows that even in a state considered undifferentiated, individual cells are not at all the same. Some may already be slightly different from others, perhaps in a way that means they're already learning or leaning towards particular development paths. By day 3 and 7, the PCA plot shows many cells beginning to drift towards new areas in PC space, which suggests that their nuclear biochemistry has started to change. And these changes might include shifts and peaks related to the amide 1 band or the cytochrome C, which could reflect chromatin being reorganized or protein expression being altered as genes involved in differentiation begin to activate. And by the time the cells reach day 10 and day 14, the points on the PCA plot become much more tightly clustered, and this indicates a more uniform and likely stable differentiated state. The authors highlight that the standard deviation in PC scores, especially alongside the first and second principal components, PC1 and PC2, it gets smaller. And this narrowing shows that the cell population is covering on fewer, more defined endpoints as it moves away from pluripotency and takes on more specialized identities. They relate this observation to the classical idea of the Waddington landscape. And in that model, stem cells start out with many possible fates, just like the broad scatter seen in early PCA plots. But then they roll down into distinct valleys or lineages as development proceeds. In my opinion, what's especially interesting is that the author's proposed Raman spec could be used to monitor these kinds of changes in real time, and this would allow researchers to identify and isolate subpopulations of cells at specific points along the differentiation path, all without needing to use any destructive labeling techniques. And this really shows how powerful Raman spectroscopy could be for continuously and non-invasively tracking how single cells or a group of cells moves from a pluripotent state into a more specialized one. And for stem cell biology and regenerative medicine, this kind of single cell insight could be incredibly valuable, especially when it comes down to improving culture conditions or ensuring that differentiated cells are consistent and reliable enough for clinical use. One of the strongest implications of this research is the potential for real-time quality control in stem cell cultures. Because Raman spec is non-destructive and label-free, scientists could periodically scan an embryonic stem cell batch to confirm if the cells are mostly pluripotent or if some are drifting prematurely into differentiation. This has direct relevance for regenerative medicine, where precise control of stem cell fate is essential. Another implication is single cell heterogeneity assessment. Uh, Rather than analyzing bulk RNA or protein levels, Raman lets us see if a subset of cells might deviate from the main population, and this can affect the uniformity of differentiated products. Uh, in the article, the authors hint at synergy with system biology approaches, and they note that measuring real-time biochemical signals can complement gene expression or epigenetic data. And this synergy would sharpen our understanding of how specific molecular events, like changes in histone marks, manifest as it shifts in Raman peaks. Finally, the technique might prove useful in drug discovery, especially in screening how candidate molecules influence cell differentiation pathways, since repeated scans of the same culture can reveal shifts in spectral signatures early on, well before morphological changes become visible. So, some of the strengths that I learned about in this paper of Raman spec are how it circumvents the needs for fluorescent dyes or deconstructive labeling. And because this study examined living cells, uh, it allows the researchers to maintain the culture and continue using it after the measurement, which is an advantage that stands out compared to methods requiring fixation or cell lysis. Another strength that I found was focusing specifically on the nucleus, which is the main site of chromatin rearrangement, transcription factor activity, and other hallmarks of differentiation. And this nucleus centric approach increased the likelihood that subtle changes in protein and DNA organization would translate into detectable spectral shifts. On the computational side, PCA is a robust tool for high dimensional data, and the author's consistent preprocessing strengthens confidence in the observed cluster separations. Also, the paper demonstrated versatility by applying the method across the spectrum of cell lines, fully differentiated, partially differentiable, and truly pluripotent in those variations, and which validated that the approach is not limited to a single cell or a type of narrow conditions. And all taken together, these attributes really show how Raman plus PCA can serve as a reliable life cell assay for monitoring progression in real time. So we can't talk about the strengths without discussing the limitations. First of all, even though Raman spec can note specific peaks, for example, the amide 1 peak at 1660 or the cytochrome C peak at 1580, they don't definitively prove the detailed biochemical events behind each principal component. And this is partly because Raman is inherently a global signal which captures many molecules simultaneously, and more targeted methods like fluorescence imaging of individual nuclear components or mass spec of extracted materials would be needed to pinpoint which epigenetic or protein changes directly drive the PC cluster shifts. Second, throughput is very slow, and it's slower than flow cytometry or bulk transcriptometry, gritomics, which makes large-scale population screens challenging. Third, while the PCA plots do show distinct clusters, there is a noticeable overlap, so borderline cells might be misclassified unless a secondary marker or repeated measurement clarifies their state. And finally, it is very important to understand that the authors present snapshots at discrete time points. Continuous observation of the exact same cell over multiple days was not performed, even though that is one of the Raman's potential strengths. Therefore, implementing a truly longitudinal single-cell time-lapse approach remains an exciting but still unfulfilled possibility, one that would help validate the method's power to track differentiation in real time without sacrificing any of the cells. So now finally, moving forward, others in the field can see enormous promise in combining Raman with high throughput systems and advanced molecular analysis. Microfluidic Raman activated cell sorting, or also known as RACs, could one day classify and separate live cells based on their spectral signatures in real time. Pairing single cell RNA sequences or CHIP sequences with Raman data would help pinpoint the precise molecular drivers behind each principal component, linking spectral changes to gene expression or epigenetic states. Additionally, actual longitudinal tracking of individual ESCs could reveal fine-grained changes in real time. And if these enhancements do succeed, we could possibly have a powerful method for controlling and monitoring cellular differentiation in regenerative therapies. Thank you for listening to my presentation, and these are the sources that I used.